So just continuing where we got cut off in the last video, leukemia patients being treated with chemotherapy suffer from a condition called neutropenia, characterized by a shortage of neutrophils, because the chemotherapy is targeting their bone marrow and killing cells in the bone marrow to try and destroy the cancer cells that are causing the leukemia. But because so much of the bone marrow activity is dedicated to producing neutrophils, because a lot of these bone marrow cells are being killed, we're not getting as many neutrophils as we need being produced. So this immunosuppresses leukemia patients. So they have very weak immune systems because these neutrophils are very important. They're like, if you want to imagine macrophages, neutrophils have very similar function to macrophages. But just think of neutrophils as kind of like a cheaper kind of soldier. So think about these as just like your foot soldiers, your basic infantry. Then macrophages, they're still common, but they're a lot larger, they're more long-lived, they're kind of more like the captains or lieutenants of the innate immune system rather than these just basic infantry troops. So moving on to mast cells, they're generally found in the connective tissues and the mucosal membranes. They're rich in granules containing histamine and prostaglandins. Degranulation of these granules will lead to vasodilation, so making capillaries and veins more permeable, so that makes diapedesis easier. So remember we said before about macrophages and dendritic cells being able to leave the blood through diapedesis. And neutrophils. So we're getting the macrophages, the dendritic cells, and the neutrophils being able to leave the blood and enter the site of infection through diapedesis, because these mast cells hanging around in these connective tissues and mucosal tissues, they're releasing these histamine and prostaglandins, which are making the capillaries and veins more permeable. So the macrophages, dendritic cells, and neutrophils are able to leave the blood and deal with any pathogens at this site of infection that the mast cells have picked up. And we also get increased endothelial permeability, which basically does the same thing. It just allows them to move freely through the endothelium. So vasodilation, making it easier for these innate immune cells to get out of the blood and into the tissues, and then increased endothelial permeability, easier for these innate immune cells to travel through the tissues to the site of infection. Mast cells mainly deal with parasitic infections as well as allergic reactions, because they're located in the connective tissues and mucous membrane. So that's why if you're allergic to pollen and you get it into your nose, you have mucous membranes in your nose containing these mast cells. But your mast cells, they have they're being receptive to harmless antigens that come from the pollen, because you've had some kind of error where we've seen B cells that are capable of producing antibodies that are receptive to these pollen antigens. These antibodies eventually get picked up by mast cells, and then that's how we get allergic reactions. We'll go deeper into it when we go over the 4 types of hypersensitivity reactions a bit later in the playlist. So the specific type of antibody that we see on the mast cell membrane. So if you look at the bottom right, we can see the mast cell, and it's got these little blue things that look like the letter Y. They're antibodies. The specific type of antibody, they are immunoglobulin E antibodies covering their membrane. When these immunoglobulin E antibodies detect an antigen they're complementary to, this triggers degranulation of the mast cell, and then the degranulation is going to release these histamine and prostaglandins, triggering the inflammatory response and vasodilation, basically causing the symptoms of the allergic reaction, which can either be minor, like in hay fever, or quite severe, if you want to think about stuff like anaphylactic shock. So allergic reactions occur when immunoglobulins with variable regions, which are these little tips of the top part of the Y shown in the picture with the IgE in yellow. If these variable regions are complementary to antigens on harmless antigens that come from stuff like pollen, then the mast cell doesn't know that. It just has these immunoglobulin E on its membrane. It thinks it's detected a dangerous pathogen, so it's going to trigger this immune response, which is why we have allergic reactions and runny noses in the spring. If you have these immunoglobulin E that are complementary to pollen antigens. So the last cell we're going to talk about for the innate immune system, this is the only one that we've talked about that doesn't come from the myeloid stem cell lineage. It comes from the lymphoid stem cell lineage. These are the natural killer cells. They're mainly found in the blood, and they play a role in bodily defenses against viruses. They trigger cellular apoptosis when they detect a pathogenic antigen on a molecule called MHC class 1 that's found on the cell membrane of lots of cells in the body. So if the antigen is found on this MHC class 1 receptor on the cell membrane, the natural killer cell detects this through its own receptor called, I believe it's T cell receptor, that picks it up for natural killer cells. But if the antigen is present on the MHC class 1, the natural killer cell knows that the cell that it's just picked up this antigen from is infected, because antigens present on these MHC class 1 receptors have only been picked up intracellularly. I'm not going to go in depth to why that is. We're going to go into that when we talk more about CD4 plus helper T cells, CD8 plus cytotoxic T cells, and the natural killer cells in its own kind of video on the adaptive immune system. So natural killer cells are classified as large granulocytes, with degranulation being the thing responsible for triggering apoptosis of the target cell. And I believe in this image we can see, I'm not sure what the cytokines shown in this picture that are going to trigger apoptosis of the target cell are, but you just have to know if the natural killer cell T cell receptor detects an antigen on a target cell's MHC class 1 receptor, it's going to know that the target cell is infected with a virus or a small bacteria, like for example, we talked about Mycobacterium infecting macrophages in TB. When it detects this and it knows the cell is infected, it's going to release cytokines and chemicals that are going to degrade the target cell membrane and kill the target cell. So it'll kill the stuff